Blast phase is an unfortunate consequence of MPNs. Very often we see alterations in P53, including mutations, but also loss of 17p. This is highly problematic because this does lead to resistance to traditional anti-leukemic therapies. One of the things we've identified is that the presence of P53 mutations leads to an erythroid-biased hematopoiesis in murine models, but also there is evidence from human studies using RNA sequencing. So this begs the question, why is this occurring, number one, but number two, is this something that could be exploited therapeutically? And so we investigated this by looking at a protein called LSD1, which is involved in the regulation of erythropoiesis. And as it turns out, the gene that encodes for LSD1, KTM1A, is actually upregulated in transformation. So using an LSD inhibitor called CPI-482, we were able to show in a murine model that you can actually improve survival in leukemic mice that were JAK2 and P53 mutant, and that there wasn't a differentiation that was observed whereby you see a decrease in the erythroid cells, but actually an emergence of myeloid cells.